This small lizard escapes predators by diving underwater and repeatedly breathing from a silver bubble attached to its snout.
Water anoles inhabit tropical stream margins, where dense vegetation, wet rocks, and moving water provide immediate underwater refuges. Their streamside habitat places branches, roots, and climbing surfaces directly above pools, channels, and concealed submerged shelter.
Perching close to water lets the animal disappear from danger within seconds, before many terrestrial hunters can react.
Birds, snakes, and other predators create intense pressure for rapid escape behavior along exposed rainforest stream banks. Before diving, the lizard evaluates distance, cover, current, and the nearest safe route beneath the water safely.
A sudden leap carries the anole below the surface before most land-based predators can follow or accurately track it.
Once submerged, depth, darkness, ripples, and visual distortion make the small reptile surprisingly difficult to locate from above.
Strong toes and curved claws help it remain anchored against flowing water while hiding beneath rocks or roots.
The extraordinary respiratory behavior begins when air leaves the nostrils and forms a bubble instead of floating away. Hydrophobic scales help exhaled air remain connected to the face, keeping the bubble accessible beside both nostrils.
Semi-aquatic anoles possess scale features associated with stronger water repellency and more reliable underwater bubble retention.
The nasal bubble may connect with a broader reflective air layer trapped against hydrophobic scales across the head.
Air moves through the nostrils while the mouth stays closed, preserving a compact respiratory circuit beneath the surface.
The lizard inhales from the bubble, exhales back into it, then repeats the same cycle during submersion.
Every breathing cycle changes the bubble's composition because metabolism consumes oxygen while tissues continuously produce carbon dioxide.
Oxygen follows the ordinary vertebrate route from the lungs across thin tissue and into the circulating bloodstream.
Rebreathing extends the usefulness of existing lung air, rather than creating an entirely new respiratory organ underwater. Measured oxygen decline inside the bubble confirms that its trapped air actively supports the submerged animal's respiration. Repeated inhalation lets the lizard reuse air that would otherwise escape immediately toward the stream surface after exhalation. Recycling stretches the useful duration of a limited air store, while the animal remains concealed during a defensive dive. Each extra underwater minute increases the chance that a searching predator leaves before the lizard must resurface. Staying below the surface allows currents, shadows, and ripples to erase the original location of the sudden escape. Recorded submersions vary greatly because body size, stress, temperature, habitat structure, and individual decisions all influence duration. Some semi-aquatic anoles have remained underwater for roughly 8 minutes, although most defensive dives are considerably shorter. A maximum observation reveals biological capacity, not the routine dive length of every animal under natural conditions.
Body size, water temperature, current speed, and disturbance can all change how long a submerged lizard remains hidden.
Smaller and larger animals carry different lung volumes, metabolic demands, and bubble surface areas relative to body mass. Remaining nearly motionless lowers oxygen consumption, allowing the bubble's limited respiratory value to last substantially longer under threat. Sheltered positions behind roots and stones reduce physical effort while hiding the lizard from movement above the water. Calm pockets behind obstacles demand less energy than exposed current, where the animal must constantly resist downstream force.
Moderate flow can refresh gases around the bubble without tearing the delicate air layer away from the snout. Carbon dioxide readily dissolves into water, creating a possible route for respiratory waste to leave the bubble. Because carbon dioxide is highly soluble, surrounding water may absorb it faster than oxygen disappears from the reservoir. An exposed bubble water boundary could therefore remain useful longer than a completely sealed pocket containing the same air.
Metabolic carbon dioxide travels from active tissues through blood toward the lungs, even while the lizard remains submerged. Exhaled carbon dioxide enters the nasal bubble, where some molecules may diffuse outward into the passing stream. Carbon dioxide removal is physiologically plausible,
but recycled stored air remains the mechanism supported most directly by experiments.
Underwater rebreathing combines ordinary lungs, hydrophobic skin, trapped air, and a dynamic gas boundary beside the nostrils. The respiratory loop begins with oxygen already stored inside air, carried beneath the surface during the initial dive.
Bubble oxygen concentration falls predictably while a living lizard continues breathing repeatedly from the attached reservoir underwater.
Researchers compared that decline with artificial bubbles held underwater under similar physical conditions, but without breathing animals.
Control bubbles retained oxygen more consistently, demonstrating that biological respiration caused the stronger decline seen with lizards. Those measurements confirmed that the nasal bubble functions as an extension of the animal's existing pulmonary air supply. Oxygen enters the lungs during inhalation, while mostly unused nitrogen returns to the bubble during the following exhalation. As oxygen decreases, nitrogen occupies a greater fraction of the remaining bubble, and the respiratory mixture becomes progressively poorer. Eventually, available oxygen falls below a useful level and contributes to the lizard's decision to return toward air. The animal releases its underwater grip, swims upward, and breaks the surface to obtain a completely fresh breath. At the surface, the depleted bubble merges with open air, while the lungs refill within a single respiratory cycle. After climbing onto a nearby rock, the lizard restores normal oxygen delivery while monitoring the streamside environment for danger. Heart rate, breathing, and movement gradually return toward ordinary levels as the animal recovers between defensive dives. Another predator may appear before recovery finishes, forcing the lizard to repeat its underwater escape under greater physiological pressure. A second dive creates another nasal bubble immediately, showing that the behavior is repeatable whenever danger returns.
Every resurfacing restores a fresh gas mixture, replacing the oxygen-poor reservoir produced during the previous submerged interval. Across a single day, repeated cycles of diving, rebreathing, resurfacing, and recovery may occur around the same stream refuge.
Underwater movement may also support exploration or foraging, although predator avoidance remains the best established ecological function. Submerged stones, roots, and leaf pockets contain potential prey, as well as concealed resting places away from terrestrial hunters. The lizard balances underwater opportunity against limited oxygen, physical effort, and the continuing need to return for atmospheric air.
Scientists tested the bubble's actual respiratory value by experimentally preventing its normal formation during controlled defensive dives.
Hatched lizards entered identical water chambers, differing mainly in whether the familiar nasal air bubble remained available. Control animals preserved their ordinary hydrophobic air interface and continued cycling exhaled gas beside the nostrils underwater. A temporary surface treatment disrupted bubble attachment without changing the chamber's water temperature, depth, or overall handling conditions. After treatment, exhaled bubbles detached from the face and floated upward before the lizard could inhale them again. Those animals relied mainly on their initial lung supply, because reusable gas no longer remained beside the nostrils. Lizards retaining normal bubbles stayed submerged substantially longer than animals whose external air interface had been disrupted. In that experiment, ordinary rebreathing increased average dive duration by approximately 32% under otherwise matched conditions.
Comparable body sizes, chambers, and handling procedures strengthen the conclusion that access to the bubble caused the difference.
The result demonstrated that the air pocket performs a functional respiratory role, rather than merely reflecting trapped surface air. However, the experiment did not show permanent underwater breathing, unlimited oxygen renewal, or complete independence from atmospheric air. The bubble remains a finite reservoir, whose volume and gas composition continue changing throughout every submerged breathing cycle. Recycling improves how the initial air supply is used, but it cannot prevent eventual oxygen depletion during long dives. Possible carbon dioxide loss across the bubble surface may further delay the point when resurfacing becomes physiologically necessary. Gas may move in both directions across the interface, although each proposed exchange process requires separate experimental evidence.
Confirmed rebreathing, plausible carbon dioxide removal, and emerging oxygen uptake evidence therefore occupy different levels of scientific certainty. Successive studies progressed from behavioral observation toward gas measurement, bubble disruption, and controlled environmental experiments over several years. Natural dives remain essential because laboratory mechanisms evolved to solve ecological problems along predator-filled tropical streams.
In the wild, bubble-bearing lizards hide beneath ledges and roots, while threats continue searching along the bank above.
An extended dive allows danger to move farther away before the concealed animal cautiously returns to the surface. Comparative research revealed that underwater bubble rebreathing appeared independently in several distantly related semi-aquatic anole lineages. Separate evolutionary branches developed remarkably similar diving behavior, despite lacking one recent bubble-breathing common ancestor within Anolis. Countryside species from different American regions encountered comparable combinations of predators, water access, and underwater shelter near flowing water. Those recurring ecological pressures repeatedly favored escape strategies that turned nearby streams into temporary defensive refuges during attacks. Across independent lineages, threatened lizards entered water and formed accessible nasal bubbles using similar behavioral sequences under threat. Ordinary anole skin already possessed water-repellent microstructure before specialized underwater rebreathing evolved within semi-aquatic species. Natural selection likely co-opted that existing surface property, giving hydrophobic scales an additional respiratory function underwater. Water repellency may originally support self-cleaning or surface protection, while later becoming useful for stable bubble retention. Semi-aquatic forms maintain more persistent external air films that remain attached during movement beneath the water surface. The accessible air concentrates near the nostrils, allowing repeated inhalation while the lizard keeps its mouth closed underwater. Different species vary in bubble size, attachment area, head shape, and the number of threads within their air layer. Convergent solutions still reflect each species' body dimensions, scale texture, habitat, and preferred underwater hiding behavior underwater. Local stream conditions can determine which bubble geometry and diving strategy produce the greatest survival advantage locally. Fast rocky channels and slow shaded pools impose different costs through current strength, oxygen availability, and concealment. Calm water reduces the energy needed to hold position, though surrounding gases may refresh differently than in strong flow. Water containing more dissolved oxygen creates stronger conditions for oxygen to move toward an attached underwater bubble. Researchers therefore compared dive duration across chambers containing low, intermediate, and high dissolved oxygen concentrations in controlled trials.
Lizards remained submerged longest in the highest oxygen treatment, when water movement was kept relatively low and controlled.
Separate experiments manipulated dissolved oxygen and flow independently, preventing one environmental variable from masking the other's effect.
With oxygen held constant, slower flow treatments also produced longer dives than faster-moving water inside the experimental chambers.
The combination of adequate dissolved oxygen, low physical effort, and a stable bubble suggested an additional respiratory possibility.
A true physical gill would draw some dissolved oxygen from surrounding water into the bubble during submersion.
Aquatic spiders and insects already use air films or bubbles that exchange respiratory gases directly with water. Across such interfaces, oxygen can enter from water, while carbon dioxide leaves through the same exposed boundary.
The anole bubble changes volume and shape during dives, potentially recording several simultaneous gas movements across time.
Closed reservoir and physical gill models predict different oxygen trajectories, bubble volumes, and achievable underwater durations for the animal. Longer dives in oxygen-rich water match one important prediction expected from partial physical gill activity underwater. However, researchers have not yet measured continuous oxygen flux directly across the bubble's surface during natural rebreathing. Future microsensors could monitor gas movement at the interface without disturbing the lizard's behavior or hydrophobic air layer.
Simultaneous oxygen, carbon dioxide, and volume measurements would reveal how storage, waste removal, and replenishment interact underwater.
Portable field sensors can connect controlled respiratory mechanisms with temperature, flow, and dissolved oxygen in occupied streams. Wild lizards frequently perch near stream sections offering manageable current, adequate oxygen, and nearby underwater cover during danger.
Roots, ledges, and cavities provide better concealment than open banks, even when respiratory conditions are otherwise similar.
Seasonal rainfall, sediment, temperature, and vegetation can alter both the quality of refuges and bubble performance throughout the year. Warmer streams hold less dissolved oxygen while simultaneously increasing the lizard's metabolic demand during underwater hiding at the same time. Shaded riparian forest may preserve cooler, oxygen-rich channels better than exposed streams altered by canopy loss. Bubble breathing, therefore, depends on an intact combination of stream structure, water chemistry, vegetation, and predator ecology. Repeated breathing, oxygen decline, bubble removal effects, and dissolved oxygen experiments now support several connected parts of the mechanism. Evidence is strongest for recycled air, while direct oxygen replenishment from water remains an active research question. A tiny nasal bubble helps this lizard stay hidden by combining stored air, gas exchange, and remarkable surface biology. This Antarctic fish reaches adulthood without red blood cells, leaving its circulating blood pale and nearly transparent. Its entire evolutionary story unfolded within the isolated, freezing waters surrounding Antarctica. The Antarctic circumpolar current helped isolate southern fishes from warmer oceans for millions of years, supporting southern ocean separation. These seas remain cold enough to freeze the body fluids of most unprotected fishes, central to sub-zero seawater exposure. Antifreeze glycoproteins prevent dangerous ice crystals from growing inside many Antarctic nodovoid fishes, central to antifreeze crystal control. These proteins attach to ice and interrupt the repeating structure needed for further growth, supporting ice-binding molecular defense. Cold water slows biochemical reactions, movement, digestion, and nearly every other process inside the animal, supporting cold-slowed biochemistry. A comparatively low metabolic demand reduces the amount of oxygen its tissues must receive each minute, supporting reduced metabolic demand. Nearly every other vertebrate transports oxygen using red cells packed with hemoglobin, while preserving standard vertebrate oxygen transport. Hemoglobin multiplies blood's oxygen capacity by reversibly binding gas molecules inside circulating cells, which affects hemoglobin oxygen binding. Icefish blood lacks both the red cells and their dominant respiratory pigment, sustaining translucent plasma circulation throughout Antarctic activity. Under a microscope, its circulation appears strikingly different from ordinary vertebrate blood, which affects erythrocyte-free blood samples. Oxygen must travel physically dissolved within plasma rather than attached to hemoglobin, sustaining plasma-dissolved oxygen throughout Antarctic activity.
That method carries only a small fraction of the oxygen transported by red-blooded relatives, supporting weak oxygen delivery.
Losing hemoglobin reduced blood oxygen capacity by roughly an order of magnitude, with consequences for collapsed carrying capacity. Yet the fish survives because its entire circulatory system compensates for that enormous deficit, supporting whole system compensation. No single adaptation explains life without red blood. Several mechanisms work together continuously, which cold seawater naturally holds more dissolved oxygen than warm seawater, compensated by cold water oxygen abundance inside Antarctic tissues. Abundant environmental oxygen improves diffusion across the gills, despite weak transport inside the blood, which affects gill diffusion efficiency.
The Antarctic environment made a normally catastrophic blood defect temporarily survivable.
Adult crocodile icefishes are the only known vertebrates completely lacking circulating red blood cells, central to adult erythrocyte absence. Their developmental program no longer produces the mature oxygen-carrying cells typical of vertebrates, supporting arrested blood cell development. Genomic evidence records the breakdown of genes required for functional hemoglobin, compensated by globin gene degeneration inside Antarctic tissues. Key globin genes were deleted or reduced to non-functional genomic remnants, compensated by non-functional genomic remnants inside Antarctic tissues. Globin loss occurred after the icefish lineage separated from hemoglobin-producing notothenoids, while preserving post-divergence hemoglobin loss. Modern white-blooded icefishes inherited the condition from a common ancestor, compensated by shared ancestral deletion inside Antarctic tissues. The family later diversified while retaining its unusual blood physiology, compensated by icefish family diversification inside Antarctic tissues. Myoglobin followed a different evolutionary path and disappeared independently in several icefish species, which affects independent myoglobin loss.
Where present, myoglobin helps buffer oxygen inside constantly active heart cells, revealing cardiac oxygen buffering under stable cold conditions.
Some icefish hearts function without either hemoglobin or cardiac myoglobin, sustaining myoglobin-free heart function throughout Antarctic activity. Independent losses demonstrate that similar physiology evolved repeatedly within the family, compensated by repeated evolutionary loss inside Antarctic tissues.
Scientists still debate whether hemoglobin loss was advantageous or merely survivable under extreme cold.
Removing red cells made blood easier to pump while severely weakening oxygen transport, which affects viscosity-capacity trade-offs. Lower viscosity reduces resistance within the circulation, partly offset through low-resistance plasma flow across cold tissues. Greater cardiac output partly offset the small amount of oxygen carried per unit blood, which affects elevated cardiac output.
Wide vessels allow high-volume flow without excessive pressure, partly offset through wide vessel circulation across cold tissues. Dense capillary networks shorten the distance between plasma oxygen and tissue cells, tied closely to dense capillary architecture. Mitochondrial abundance increases the cellular surface available for oxygen use, partly offset through mitochondrial oxygen use across cold tissues. Icefish mitochondria also display unusual density, size, and internal organization, partly offset through unusual mitochondrial morphology across cold tissues.
Hemoglobin loss reshaped anatomy from genes to vessels, heart, and cellular energy machinery, especially through genome-to-heart compensation.
Every delivered oxygen molecule begins as gas dissolved within the surrounding seawater, while preserving gill plasma oxygen transfer.
Lower temperature increases the amount of oxygen water can hold, compensated by temperature-dependent oxygen solubility inside Antarctic tissues. Polar conditions therefore maximize the oxygen available for passive uptake, compensated by polar passive uptake inside Antarctic tissues. Thin exchange surfaces reduce the distance oxygen must diffuse, partly offset through thin membrane diffusion across cold tissues.
No cellular carrier captures the gas after it enters circulation, revealing carrier-free circulation under stable cold conditions.
Carrying more plasma increases the total dissolved oxygen moving through the animal, sustaining expanded blood volume throughout Antarctic activity. Icefishes maintain substantially greater blood volume than many related fishes, partly offset through high plasma volume across cold tissues.
Cell-free blood flows with comparatively low resistance, partly offset through low cellular crowding across cold tissues.
High vascular density increases the area available for oxygen delivery, compensated by vascular oxygen delivery area inside Antarctic tissues. Short diffusion distances compensate partly for low oxygen concentration, partly offset through short tissue diffusion across cold tissues. Delivery succeeds through broad distribution rather than concentrated oxygen cargo, partly offset through distributed capillary delivery across cold tissues. Behavior also keeps oxygen demand within the circulation's restricted capacity, partly offset through behavioral oxygen conservation across cold tissues. Intense activity can challenge a system with little oxygen reserve, partly offset through burst swimming limitation across cold tissues. Icefishes generally possess limited scope for prolonged high-performance swimming, partly offset through low aerobic scope across cold tissues. Warmer conditions raise metabolic demand while lowering water's oxygen content, compensated by warming-driven oxygen mismatch inside Antarctic tissues. Temperature therefore compresses the animal's physiological safety margin from both directions, compensated by shrinking physiological margin inside Antarctic tissues. Its unusual blood works best within the environment where it evolved, compensated by stable sub-zero performance inside Antarctic tissues.
Depth and local circulation can provide relatively stable thermal conditions, compensated by deep water thermal refuge inside Antarctic tissues.
Shallow populations may encounter faster environmental change through shallow water exposure, limiting stress while sustaining water exposure.
Environmental oxygen and cardiovascular compensation remain inseparable parts of the same survival system, which affects environment circulation coupling. Icefish hearts are enlarged relative to those of similarly sized red-blooded relatives, which affects enlarged ventricular anatomy. Each beat moves an unusually large volume through the circulation, supporting high stroke volume throughout Antarctic cardiac activity. High stroke volume raises cardiac output without requiring extremely rapid heart rates, with consequences for cardiac output compensation. Old cardiac muscle contracts slowly and depends on specialized cellular machinery, compensated by cold cardiac machinery inside Antarctic tissues. Mitochondrial density supports aerobic energy production inside the oversized heart, partly offset through heart mitochondrial density across cold tissues. Dense vessels keep oxygen pathways short within cardiac tissue, partly offset through short cardiac diffusion across cold tissues. Some species add myoglobin storage within the heart, partly offset through retained cardiac myoglobin across cold tissues. Other species survive after losing cardiac myoglobin independently, partly offset through independent myoglobin absence across cold tissues. Different lineages reach similar physiological outcomes through distinct genetic changes, partly offset through convergent cardiovascular outcomes across cold tissues.
Elevated nitric oxide signaling may help promote wide vessels and vascular growth, tied closely to nitric oxide signaling.
Nitric oxide regulates both blood flow and formation of new vessels, compensated by angiogenic vessel growth inside Antarctic tissues.
Long-term signaling can reshape anatomy throughout growth, through developmental compensation.
Vascular differences emerge as the animal develops, through heart growth trajectories, limiting stress while sustaining heart growth trajectories in Antarctica.
Static signals may transform a blood defect into a stable body plan, sustaining homeostatic circulatory remodeling throughout Antarctic activity. Low viscosity makes large volume pumping mechanically easier, partly offset through viscosity-enabled high flow across cold tissues. Tips. Cardiovascular compensation still requires substantial energy. Through energetic compensation costs, limiting the fish pays for weak blood with a larger circulatory apparatus, revealing large circulatory investment under stable cold conditions. Warmer water can push that enlarged system toward its performance ceiling, sustaining thermal cardiac limits throughout Antarctic activity. Cardiac capacity may limit acute warming tolerance in some species, partly offset through cardiac performance ceiling across cold tissues. Every compensation traces back to an ancient loss recorded in the genome, with consequences for integrated polar physiology. Their closest red-blooded relatives still retain that ancestral system, partly offset through retained relative physiology across cold tissues. A major deletion destroyed essential hemoglobin coating material, partly offset through beta-globin deletion across cold tissues. Additional remnants preserve a molecular fossil of the lost pathway, compensated by alpha-globin genomic fossil inside Antarctic tissues. Once the protein was no longer required, disabling mutations accumulated more freely, central to post-loss mutation accumulation. Blood physiology changed far beyond the globin genes themselves, partly offset through reduced iron physiology across cold tissues. White blood contains dramatically less iron bound in respiratory pigment, compensated by iron-poor white blood inside Antarctic tissues. The absence of red cells lowers viscosity substantially, partly offset through erythrocyte-free viscosity across cold tissues. Lower viscosity may have softened the damage without making hemoglobin loss beneficial, sustaining evolutionary trade-off interpretation throughout Antarctic activity. Environmental conditions created a rare window in which the mutation could persist, sustaining cold ancestral tolerance throughout Antarctic activity. Similar gene loss would be far less tolerable in warmer, oxygen-poor seas, which affects warm water incompatibility. Red-blooded relatives often retain greater capacity for oxygen transport during thermal stress, which affects relative thermal capacity. Extreme specialization can increase success in stable environments while reducing resilience to rapid change. Supporting specialization, genome studies reveal additional adaptations beyond blood and circulation, partly offset through antioxidant gene expansion across cold tissues. Oxygen-rich water and abundant mitochondria can increase oxidative stress, partly offset through mitochondrial oxidative pressure across cold tissues. Strong antioxidant and repair systems limit damage from reactive molecules, partly offset through macromolecular repair defense across cold tissues. Icefish survival depends on a broader Antarctic molecular toolkit, partly offset through broader cold adaptation across cold tissues. Traits optimized for constant cold may become liabilities during warming, partly offset through warming-created mismatch across cold tissues. Climate change modifies several ecological pressures simultaneously through multi-factor climate pressure.
Risk will vary among species, locations, depths, and life stages, partly offset through species-specific exposure across cold tissues. Warm water holds less dissolved oxygen while animal metabolism accelerates, partly offset through warming oxygen decline across cold tissues. Icefish face simultaneous reductions in supply and increases in demand, compensated by aerobic supply-demand compression inside Antarctic tissues.
Limited oxygen carrying capacity may bring some ice fishes to their ceiling sooner, tied closely to earlier performance ceilings. Thermal vulnerability is not identical across the family, partly offset through variable family vulnerability across cold tissues. Behavioral movement may reduce exposure to warming surface layers, partly offset through behavioral thermal refuge across cold tissues. Adaptations. Suitable cold refuges may not exist everywhere. Through geographic restraints, limiting stress, while early life stages may encounter different thermal risks than adults, compensated by larval thermal exposure inside Antarctic tissues. Warming can reshape development before circulatory compensation is fully established, balanced through developmental oxygen stress during polar life. Changing prey fields can indirectly affect icefish energy budgets, partly offset through changing prey networks across cold tissues. Reduced food could make costly cardiovascular compensation harder to maintain, partly offset through cardiovascular energy deficit across cold tissues. Field monitoring is essential for separating short experiments from population trends, with consequences for long-term population monitoring. Research collections reveal anatomy, genetics, diet, and physiological condition, partly offset through research specimen collection across cold tissues. Modern tools connect whole animal performance with molecular mechanisms, partly offset through integrated molecular analysis across cold tissues. Comparative studies identify which traits followed hemoglobin loss and which evolved earlier, with consequences for comparative evolutionary mapping. Variation within the family turns icefishes into a natural evolutionary experiment, with consequences for within-family physiological variation.
Biology offers insights into oxygen transport without providing a direct model for human treatment, supporting biomedical research boundaries.
Genes. Understanding compensation may illuminate general principles of cardiovascular plasticity, partly offset.
Those adaptations remain inseparable from the cold, oxygen-rich sea that made them possible, helping explain Southern Ocean dependence.
Its future depends on whether physiology can keep pace with changing Antarctic water, tied closely to future warming pressure.
Antarctic icefish survive without red blood by rebuilding nearly every other part of oxygen delivery, supporting integrated oxygen delivery.
This seabird can sleep while remaining airborne above the open ocean. Long flights inspired a question that behavior alone could not answer. Researchers studied breeding female great frigate birds from a Galapagos colony. A lightweight recorder was attached before each bird returned to flight.
Electrodes measured electrical activity from both brain hemispheres.
An accelerometer recorded head motion and turning forces.
A separate GPS unit tracked position and altitude.
Together, the instruments linked brain state with real flight behavior.
Some recorded journeys covered thousands of kilometers without landing. Longest sleep recordings followed birds aloft for about 10 days. Over deep water, the birds had nowhere safe to perch.
Frigate birds avoid prolonged contact with the ocean surface.
Their plumage is less waterproof than that of many seabirds.
Getting airborne after becoming soaked would be difficult. Low wing loading makes sustained soaring unusually efficient.
A deeply forked tail provides precise aerial control. Atmospheric updrafts supply altitude without constant flapping. Circling converts rising air into height. Stored altitude then powers long descending glides.
Soaring reduces the muscular cost of staying airborne.
direct brain recordings finally revealed whether flight contained genuine sleep. Sometimes one hemisphere entered slow-wave sleep while the other remained more awake.
Bird vision is strongly crossed between eye and opposite hemisphere.
The awake hemisphere could monitor visual information from the open eye. During asymmetric sleep, the open eye often faced the direction of turning. Visual monitoring may help avoid collisions inside crowded rising air. Half-brain sleep preserved some vigilance during flight. Unihemispheric sleep also helps other birds monitor danger on land. In frigate birds, vigilance appears linked to navigation and nearby traffic.
The hemispheres can differ in sleep depth, rather than switching completely.
Sleep exists along a continuum of hemispheric asymmetry. Frigate birds also slept with both hemispheres simultaneously. Bihemispheric sleep showed that one awake hemisphere was not always required.
Aerodynamic stability can persist through short bouts of whole brain sleep.
Updrafts reduce the need for active altitude control.
Most flight sleep occurred while birds circled in rising air.
Light provides time before a sleeping bird approaches the water.
Birds generally remained awake near the ocean surface. Foraging demands favored wakefulness during daylight. During flight, that half-brain sleep allowed rest without surrendering all visual attention. Eye state matched the turning direction during many asymmetric sleep bouts. Either hemisphere could become the deeper sleeping side. Sleep arrived in short fragments rather than long, continuous periods. Typical flight sleep bouts lasted only around 12 seconds. The longest uninterrupted episode remained under 6 minutes. Dozens of brief naps accumulated across darkness.
Sleep occupied only a small fraction of time aloft.
Recorded birds slept during less than 3% of flight.
Daily flight sleep averaged less than 1 hour.
Ecological demands kept the birds awake most of each day and night.
Frigate birds track ocean features associated with future feeding opportunities.
Following changing ocean patterns may require sustained attention.
Large predators can push prey toward the surface. Frigate birds capture food while remaining airborne.
They sometimes harass other seabirds for food.
Kleptoparasitism provides another aerial feeding strategy.
Efficient feeding must support days of continuous movement. Aerodynamics minimize the cost of remaining aloft.
Night flight involved even less flapping than daytime travel. Despite sleeping in flight, the birds obtained remarkably little sleep.
Land sleep exceeded flight sleep by more than an order of magnitude.
Back at the colony, sleep became longer and deeper. Land allowed more continuous slow-wave sleep.
Flight sleep was generally lighter than land sleep.
A secure surface removed the need for aerodynamic control.
The recorded females were returning to chicks between foraging trips.
Parental demand made long foraging journeys necessary.
Food resources could lie thousands of kilometers away.
Returning to land created an opportunity for extended sleep.
Researchers still debate how completely land sleep repays losses from flight. Available recordings do not capture every transition between sea and land. Longer studies could measure cumulative sleep recovery.
New biologgers may reveal how sleep changes during longer flights.
Tracking studies show longer airborne journeys than current brain recordings cover.
Continuous flight does not automatically prove continuous in-flight sleep.
Brain activity is required to identify sleep reliably. EEG distinguishes the major avian brain states.
Rare flight bouts included rapid eye movement sleep.
Brief REM did not visibly disrupt the recorded trajectory.
Avian REM episodes can last only seconds. Reduced muscle tone was brief enough for flight to continue. Atmospheric support may buffer momentary reductions in active control. Several safeguards overlap during these rare episodes.
REM sleep ended rapidly. Most sleep aloft consisted of slow-wave sleep.
Both asymmetric and symmetric patterns occurred.
Sleep strongly favored circling flight.
Tension remained highest near potential feeding zones.
Birds often gained height before sleeping. Flight sleep combined several strategies rather than one simple autopilot. Altitude reduced immediate danger from descent.
Thermals supplied passive support.
Unihemispheric sleep preserved directional awareness. Brief naps limited exposure to reduced attention.
Together, these features made sleep aloft possible.
Human responses cannot be inferred directly from a seabird. How frigatebirds function on so little sleep remains unresolved.
Several physiological explanations are still under investigation.
Sleep may be regulated more locally than whole brain labels suggest.
On land, the bird finally gains a stable place to rest. Both hemispheres can enter deeper sleep. Daily sleep rises to more than 12 hours. Slow wave activity becomes stronger. Adhika.
Sleep episodes become more continuous. REM sleep also occurs safely on land.
Resting conditions permit fuller sleep architecture.
Land sleep likely contributes to recovery. Complete repayment of lost sleep remains uncertain. Future studies may reveal the hidden costs of oceanic sleeplessness. Other species may use different sleep strategies. Long flight alone cannot establish sleep on the wing.
Smaller instruments could expand direct tests.
New recordings may reveal multiple solutions to sleep during migration. Great frigatebirds remain the first birds directly proven to sleep in flight. Their recordings revealed several unexpected sleep states. One eye can monitor the turn while the opposite hemisphere rests.
Sometimes the entire brain sleeps while flight continues.
Yet total sleep remains astonishingly small at sea. Frigatebirds sleep in the sky by combining brief naps, soaring, and partial vigilance.
The Great Pyramid still contains spaces whose purpose and exact shape remain unknown.
Khufu's monument belongs to a much larger royal landscape at Giza. Originally, polished limestone covered the stepped core visible today. Erosion and stone removal changed the monument's outer profile. Its builders established an exceptionally regular base.
The pyramid's orientation closely follows the cardinal directions.
Ancient surveyors had several possible methods for finding true north. The exact alignment procedure remains debated. The monument required quarrying, transport, and positioning on a massive scale. Egyptian crews relied on familiar materials, organized with extraordinary coordination. Much of the core limestone came from local quarries beside the pyramid. Workers isolated blocks by cutting trenches around them. Levers helped separate and reposition heavy stones. Sleds distributed weight across the ground. Moistening sand can reduce the force needed to pull a sled.
Water transport moved selected stone closer to the plateau. High-quality casing limestone arrived from the Tura quarries. Granite for internal chambers traveled from southern Egypt. The river linked distant quarries with the building site.
The pyramid depended on transport infrastructure as much as stoneworking skill.
The known interior begins with a sloping passage entering from the north. Part of the route cuts directly through the plateau's limestone. The subterranean chamber was left incomplete.
Its intended function remains uncertain.
A concealed junction branches upward toward the pyramid's main chambers. This route was carefully built within the rising masonry.
The Grand Gallery is the pyramid's most dramatic known internal space. Its walls narrow upward through overlapping courses. Architectural details suggest specialized construction or closure procedures. No single explanation for the gallery's full role is universally accepted.
A separate passage leads to the chamber, traditionally called the Queen's Chamber.
No queen was found there, and the traditional name may mislead.
The niche's original purpose is unknown. Small channels extend upward from the chamber. Exploration found doors or blocking stones within the shafts.
Copper-like fittings increased curiosity without revealing the space beyond.
Robotic probes exposed another confined section, not a large treasure chamber.
The upper route passes through a complex granite antechamber. Granite barriers could seal the final chamber route. The king's chamber contains the monument's surviving stone sarcophagus.
Above the chamber, stacked spaces redistribute enormous masonry loads.
The upper roof channels pressure away from the chamber ceiling. Internal geometry was engineered to manage crushing forces.
Modern scans revealed a major unknown space above the Grand Gallery.
Its precise shape cannot yet be reconstructed. The detected zone extends for at least about 30 meters.
Muon data do not yet determine its orientation or internal divisions.
Its existence is better established than its architecture. Independent muon measurements confirmed the anomaly. Cosmic particles made the hidden space visible without drilling. Another scan detected a corridor behind the pyramid's north face chevrons.
The hidden corridor extends inward for roughly 9 meters.
Its destination and function remain uncertain. A miniature camera provided the first direct images inside.
The corridor has a gabled ceiling beneath the external chevrons. One possibility is that it protects masonry below from weight.
Structural and access-related functions are both discussed.
No confirmed doorway has been found at its end.
Additional methods have reinforced the corridor's location.
Multiple techniques now describe the same hidden structure.
No proven passage currently connects the two discoveries. The pyramid's interior mixes documented corridors with unresolved anomalies.
Different discoveries carry different levels of certainty.
Scientists distinguish physical entry from remote imaging.
Voids transmit more cosmic muons than dense masonry.
Large datasets convert particle counts into internal density images. Repeated measurements reduce the chance of a false signal. The big void remained visible across independent analyses. No evidence shows that the void contains treasure or a royal burial.
Its role may relate to construction, load management, or an unknown passage system. Understanding hidden spaces may reveal how the pyramid was assembled. Internal passages had to remain accessible during construction.
Temporary construction routes could later disappear inside the core.
No surviving plan identifies the complete ramp system.
A single straight ramp becomes increasingly massive at greater height. Straight ramps remain plausible for early construction stages.
Switchback ramps reduce overall ramp length.
External spirals could obstruct surveying of corners and faces.
Internal ramp models place some hauling routes within the structure. Archaeological confirmation for a complete internal spiral remains limited. Levers may have raised blocks through shorter vertical steps. Temporary supports could hold stones between lifts. Ropes coordinated pulling, steering, and braking. Simple direction-changing devices may have multiplied crew control. Builders probably used different methods at different stages. Material size and route requirements changed with height. Some internal granite elements demanded specialized handling.
The exact lifting sequence for massive granite remains debated. Construction traces constrain theories even when they do not settle them. No exotic technology is required by the archaeological evidence.
archaeology uncovered a substantial community supporting pyramid construction.
The settlement contained infrastructure for feeding and organizing labor.
Staple foods sustained large rotating work teams. Animal remains show substantial meat provisioning.
Some workers survived serious injuries with care.
Construction depended on organized crews rather than chained masses.
Specialized roles connected quarry, river, workshop, and monument.
Mera's log records limestone transport to Khufu's construction site. The diary describes repeated deliveries rather than legendary machinery.
The project mobilized resources across the Egyptian state.
Nearby burials further identify the builders as Egyptian communities. The old image of foreign slaves building the pyramid lacks archaeological support.
Labor may have mixed rotating crews with long-term skilled personnel.
The Nile cycle helped organize people and supplies.
Modern mapping has reconstructed waterways closer to pyramid sites.
Ancient channels likely shortened overland stone transport.
The pyramid complex linked river access with elevated construction zones.
Monumental routes could serve both symbolic and logistical roles. The monument was a social system as well as an engineering project.
The date and circumstances of ancient looting remain unclear.
A later tunnel bypassed the original entrance system.